good news is today we're going to have a shorter lecture, and then we're going to go upstairs and you guys are going to be able to study for the lab exam. Okay? So we're just going to finish up with the female reproductive structures, and then that's it for today. We're going to go over female reproduction. Okay? So let's just go over some of the structures first. Now, again, I really like the idea of having you guys, do that sheet and just being able to label where these structures are, and then we can focus more on physiology for the exams. All right. So for the lecture exam, when we get together and do that right before the lab exam, I'll have a review session and I'll go over the physiology so that I really understand which physiology I'm talking about, exactly what. So anything that's function, I guess you would have to know, but anything as far as labeling things, or anything like that, you're not going to have to know. So I don't know if that's going to be confusing, but so anyway, this is the female anatomy. You have the uterus, and then from the uterus there's two uterine tubes. They used to be called the fallopian tubes, and then they end at this area where the fimbriae are. On the lab exam, I think you have, if I remember right, you have to know the different parts of the uterine tube, right? So we'll kind of take a look at that. The fimbriae end at the ovary. The ovary is where follicles are going to be produced, and the meiosis is going to be completed, so the oocyte is going to be completed, and estrogens and progesterone are also produced in there. Okay. In the uterus, then, we have the inner lining, which is the endometrium, the middle lining, which is the myometrium, and the outer layer, which is the epimetrium. The uterus comes to an end, it narrows down at an area that we call the cervix, and then there's an opening. We'll look at a different diagram to see that. The cervix opens up into the vaginal canal, okay, and then the vaginal canal opens up into this area of external genitalia that we call the vulva. Okay, so inside the vulva, we're going to see different structures, including the labia, labia minora, labia majorum, as well as the clitoris or the clitoris, and the openings into the urethra and the vaginal canal. Okay. So this is just a weird from the top looking down view. Down here. So this is just a superior view of an actual cadaver. So we're actually looking, thank you, Christy. Yeah. Looking down, here's the uterus, there's the uterine tubes, there's the ovaries, right there. Okay. One like this. So there's the uterus, uterine tubes, there's uterine tubes, there's the fimbriae, there's the ovary, there's the ovarian ligament, here's the broad ligament. So all of these ligaments you have to know on the lab. Okay, so let's look at the ovaries first. There's three main functions of the ovaries. They're going to produce the female gametes, which are oocytes. So we will look again at meiosis, and we'll look at, it's the secondary oocyte that actually gets released. It's not the mature ovum. It also secretes estrogens and progesterone, and it also secretes a hormone called inhibin. Inhibin is used in negative feedback. So it'll control FSH. So it'll control the secretion of FSH from the anterior pituitary, because you need FSH in order for the follicles to start developing. So we'll see that as well. So the ovaries are going to be suspended to the pelvic wall by the ovarian ligament and the suspensory ligament. Okay. So if we look at this, we can see here is the ovarian ligament right here. And here is the suspensory ligament out here. And the suspensory ligament has the ovarian artery and vein inside it. You can also see the broad ligament as well. Those are the ligaments that you can see on that picture. This is a picture of the ovary itself, and we're going to go through, we're going to talk about the process of the follicle developing into a tertiary follicle and the oocyte developing into a secondary oocyte. So let's look at, okay. Before we do that, here's the three different segments of the uterine tubes. The infundibulum, that's going to be the expanded funnel near the ovary. That's going to branch out into those little fimbriae, those little finger-like projections. The ampulla is the middle segment of it, and then the isthmus is the shorter segment between the ampulla and the uterine wall. So here you can see the infundibulum right here where it widens out, and there's the ampulla, and then there's the isthmus. So all of these are going to be on your lab exam. You just have to be able to identify those on a picture. Okay, inside the uterine tube, the epithelium is a ciliated columnar epithelium, because when the secondary oocyte comes into the uterine tube, the cilia move. Remember, they're on the top of the cells, and when they can move things across the top of the cell. So they're moving fluid, and that inside that fluid is that secondary oocyte. They're trying to move it towards the uterus, right? And it'll be the secondary oocyte, if it's going to be fertilized, will be fertilized in the uterine tube itself. The other thing is that the uterine tubes also have peristaltic contractions. So in addition to having the cilia, there's also peristaltic contractions where the smooth muscle are pushing the ovum closer to the uterus. Here's a picture of the cilia, so you can kind of see what they look like. Okay, now in order for fertilization to occur, that secondary oocyte must meet the sperm within the first 12 to 24 hours in the uterine tube, right? So the female ovulates, and within 12 to 24 hours, that egg would have to be fertilized. Otherwise, it's going to be just secreted out, not secreted out, but just eliminated from the body. Usually, the fertilization occurs between that ampulla and the isthmus. That's where fertilization occurs, and then that newfound cell is going to make its way into the uterus, and that's where it's going to implant once it's into the uterus in the endometrium. All right, the uterus provides for the developing embryo. Okay, so it has an endometrial layer where the embryo can implant. The embryo is, after several cell changes, the sperm and the ovum become a embryo. And then the uterus is going to help to mechanically protect it, give it nutrition, help to get rid of any waste removal. The uterus is usually, normally bends anterior in anteflexion, but sometimes women have what's called retroflexion. It's probably a little more common than what we think, and that's where the uterus bends backward instead of forward. Here's some of the suspensory ligaments of the uterus that you have to know: the uterosacral, the round ligaments, and the cardinal ligaments. I don't think cardinal ligaments are on your lab list. And then the uterine body itself, and we'll see a picture here. The uterine body has, It's the largest portion of the uterus. It ends at the isthmus, where that uterine tube begins. And the fundus, very similar to like in the heart, the fundus is the part that is superior. It's the most superior part. We're going to look at the next picture here. It's coming up in a couple of slides. The cervix is the most inferior part of the uterus, and there's an opening in the cervix which is called the external os. That opening leads into the cervical canal, and which is a constricted passageway that leads to the internal os, which is another opening that will lead into the vaginal canal. So there's some arteries of the uterus. I'm not going to go into those. All right, so here, let's see. All right, that's not a great picture. Here's the round ligament. So you will see this picture in lab. There's the round ligament right there. You can also see the ovarian ligament. You can also see the suspensory ligament and the broad ligament. This is the external os, the opening into the cervix. But again, can't really see the, there's the internal os right there, and then there's the external os right there. So you can kind of see those openings on this picture. Okay. Internal os, cervical canal, external os, vaginal canal. And this whole thing is called the cervix. Okay, the uterine wall has 3 layers to it. The perimetrium, which is the outermost layer, the myometrium, which is the middle layer, and the endometrium, which is the inner layer. The perimetrium, or the outer layer, is this, it's continuous with that peritoneal, visceral peritoneum. Okay, so the visceral peritoneum and the perimetrium are kind of connected together. The myometrium has the muscles, the muscular layer, that's what's going to contract, and then the endometrium has a thin glandular layer that changes in size, that we'll see. Okay, so the endometrium. It has many uterine glands in it, and estrogens are going to cause the uterine glands to change monthly. So estrogen levels, there's a thin, like, functional layer of the endometrium, and when estrogens are produced, it's going to make that lining thicker. And progesterone, when it gets secreted, helps to keep that lining thicker. Okay. So this is kind of a picture of it here, the endometrium, right in here. Okay. And the functional layer of it is always going to be sustained. So you always have one layer of cells that are stem cells, and they're always going to be there. They don't get shed off. But then the other layers that are produced each month, they get shed at the time of ovulation, or at the time of menstruation. So here's this, the basilar layer, the functional layer. You see some uterine glands and some simple epithelium here. Okay, so we have the functional layer, which contains most of the uterine glands, contributes to most of the endometrial thickness. It's going to be the one that gets thicker and then sheds, right? But one part of the layer is going to always remain. The basal layer is the deeper layer, and that's going to have some of those more terminal tubular uterine glands. So it's just kind of, it just keeps going over the same thing that I had said, that it's just going to change on a month-to-month basis. And we will talk about the cycle in just a minute. All right, as far as the vagina goes, the vagina is a muscular tube as well. Okay, this muscular tube is, it's a passageway for the the lining of the endometrium as it sheds each month. It's a passageway to eliminate the shedding. It's also a passageway to receive the sperm during sexual intercourse. And it also forms the most inferior portion of the birth canal. The cervix is going to project into the vaginal canal, and then there's a shallow recess that surrounds, just surrounds that cervix, which is called the vaginal fornix, and I believe that's on your lab list as well. So the vagina is just, it's parallel to the rectum. It's posterior to the rectum, and it's just in front of, it's just in front of the rectum and just behind the urethra. That's the way that says, it's kind of weird. There's two different muscles on either side of the vaginal entrance. One is the bulbospongiosus muscle. Let's see, I think I'm really on here. Let's just go right here so we can see it. You guys, I know you've probably heard of the hymen. The hymen is an elastic epithelial fold that partially blocks the entrance to the vagina. It's usually torn early in a young adolescent girl, usually by if tampons are used. Otherwise, it gets torn during sexual intercourse, pelvic examination, sometimes physical activity. These types of things can also tear the hymen, but I don't have a picture of that either, so I apologize for that. All right, so that's that. Okay, the external genitalia then is called the vulva, right? So the whole area is called the vulva, that area. There's a vestibule that's surrounded by these labia. You have labia minora. This is the innermost layer of skin that doesn't have any hair on it. And then there's the labia majora, which is the outer layer of small folds of skin, and that does have hair on it. I'm just so not used to looking at words here I'm talking about. I feel like I'm just reading off of here. You guys remember all this stuff, right? From general? There's two urethral glands that discharge into the urethra near the external opening. I'll show you where those are. You're going to have to be able to identify those in lab. The clitoris, or the clitoris, however you want to say it, that's the erectile tissue that's similar to the erectile tissue in the male. Okay. Let's see if there's anything else. All right, then the outer part, I already told you about the labia majora. So here's a picture of the female genitalia. This is the vulva. So the outermost area has the hair on it, that's the labia majora. The skin on the inside, that's called the labia minora. The vestibule is the area between the two sides of the labia minora. The hymen here, you're seeing, is torn. It's a thin piece of skin that's closed, but as soon as there's any type of projection in there, it's torn. So this one's showing a torn hymen. Let's see. Here's the greater vestibular gland. This is the urethral opening. This is the prepuce on the female, is just a little piece of skin that's overlying the clitoris. And then there's the glands of the clitoris. So that's just external genitalia on a female. As far as the mammary glands go, the mammary glands are going to produce milk. They lie in adipose tissue that's in the pectoral area. The nipple and the areola cover, the areola is the brownish colored skin around each nipple, and then the nipple contains the ducts of the mammary glands. So, all right, so looking at the picture, here's the areolar area. This is the darkened area. There's the nipple. Here's some of the ducts, the lactiferous ducts leading into the lobules of the mammary gland. Okay, we can see that they're separated here into lobules by the suspensory ligaments, and there's adipose tissue surrounding all of those. And they say pectoral area because you can see the pectoralis major muscle is right behind it. Okay, all right, so that's a lot of the structure, all right? And you know what you need to know based on what we have in the lab exam, or lab list. Okay, so now let's talk about the process of producing the process of oogenesis. Now, it's very similar to the mitosis and meiosis of spermatogenesis, but there's some major differences. First of all, in spermatogenesis, those stem cells, those spermatogonia, they're not going to be stimulated to produce the follicles or to produce the spermatozoa until puberty, right? So all of that starts at puberty in the male. Spermatogonia, all the way to the spermatozoa. That's going to happen in puberty. In the female, oogenesis begins before birth. So we're going to see that before birth, those oogonia are going to become primary oocytes, and then they halt there. Okay, so it starts before birth and it halts as a primary oocyte. And then it will continue going through meiosis once the girl gets to puberty. Once she gets to puberty, then meiosis 2 begins, and that primary oocyte becomes a secondary oocyte, but it halts in secondary oocyte right in the metaphase 2 part. And then that's where it's going to be ovulated. And then a sperm will have to fertilize that secondary oocyte before it would complete meiosis 2 to become what we call an ovum. All right, so we're going to have to look at all of that. So oogenesis, unlike the male, it begins before birth, it accelerates at puberty, and then it's going to end at menopause. Where in the male, theirs is not going to end. That spermatogenesis isn't going to end that early. Okay. All right, so let's look at the female ovaries. They contain those stem cells, which are called oogonium. Okay? The oogonium are going to go through mitosis. At the end of mitosis, they're going to produce primary oocytes. Those primary oocytes are identical to the stem cell. They have 23 pairs of chromosomes. This 2N is just telling you that it's a diploid, okay, 2N meaning diploid. So there's 23 pairs of chromosomes. And about 7 million of those primary, we're going to see how the primary oocytes, about 7 million of those primary oocytes are going to be present after 5 months of development, right? And then that's what she's born with. She will be born with that many primary oocytes, right? So we should take a look at mitosis. This should be very familiar with you. Here we started out with a stem cell. We had 23 pairs of chromosomes. Went through mitosis, prophase, anaphase, prophase, metaphase, anaphase, telophase. At the end, we ended up with 2 identical cells that are identical to the first cell. Each one of them have 23 pairs of chromosomes. Okay? So my mitosis occurs before birth. And then meiosis 1 begins before birth, before that baby girl is born. So we look at this, we started with an oogonium. Okay? The oogonium is going to become primary oocytes. Now, you know, the other difference in the male, that spermatogonium, one of them stayed as a stem cell, the other one became the primary oocyte. In this case, she's going to have all she's going to need at birth, so both of those oogonium are going to become primary oocytes. Right? This primary oocyte, each one then, is going to go through the process of meiosis 1, okay? And at the end of meiosis 1, and actually what will happen is that in meiosis 1 we have a lot of the same changes that we saw in spermatogenesis. So in prophase 1, do you remember what we got in what happened in prophase 1 that was so significant? We had tetrad formation. The tetrad forms. We had crossing over. Remember all of that? Okay. So we have all of that, and then it stops. Right here. In prophase 1. It stops. And that's what she's born with. And she's born with those 7 million primary oocytes that are locked into prophase 1. Okay? That's what she's born with, 7 million of those. All right, so now she's born. Then, okay, then what happens is that there's a lot of degeneration of these primary oocytes, so that she ends up with only 2 million. Okay, they degenerate. By puberty, they keep degenerating. She'll only have about 400,000, because she's not going to end up with more than, what's the most amount of kids anyone's ever had? 16, 18, you know? She's not going to need all those 400,000 primary oocytes, all right? But that's what she will end up by the time puberty comes. Many of those will have degenerated to the point where she has only 400,000 only, all right? So now she has 400,000 primary oocytes, and only some of those will be stimulated to continue on with meiosis 1, right? And okay, and at the end of meiosis 1, so she'll continue then through metaphase 1, anaphase 1, telophase 1, and she will end up with a secondary oocyte. Okay, this is in the ovaries, all right, so every month she'll have a new group, and we'll go over this schedule too. She'll have a new group of primary oocytes that are going to be stimulated to continue on to become secondary oocytes. So all of meiosis 1 will continue. She'll end up with a few secondary oocytes. And then during I'm just going to give you the rough lowdown, and then we're going to go back and look at details. And then the secondary oocyte is going to start the process of meiosis 2. starts the process of meiosis II, goes through prophase II, okay? Goes through metaphase II. All right. Now what's happening here is the tetrads had separated up here, and the maternal gene and its copy were over here, and the paternal gene and its copy were over here. And then in this meiosis II, then the chromatid splits into separate chromosomes, daughter chromosomes. So now we're going to have a cell that's left with 23 chromosomes. And I'm going to go through this process with you guys so you can see what that looks like. But now there's 23 chromosomes locked into metaphase II. Locked into metaphase II. This is what's going to be released during ovulation. Okay? The secondary oocyte locked into metaphase II. That's what gets released during ovulation. All right, so let's go back and look at some of the details here then. Okay, so here is meiosis I. There's some interesting differences between this and spermatogenesis. All right, so here we have our primary oocyte. The primary oocyte is going to start meiosis I. At the end of meiosis I, which will be halted in prophase I until puberty, but once puberty starts, continues on. At the end of meiosis I, you end up with one secondary oocyte that is haploid. It contains the chromosome, the chromatid, basically, the paternal, you end up with a chromosome and its copy. 23 chromosomes plus their copy. And the chromosome and the copy are linked together. So you have a secondary oocyte, and where we would normally see the primary oocyte when it goes through meiosis, we saw in spermatogenesis, it ended up with 2 secondary spermatocytes. One of the oocytes, one of the secondary oocytes doesn't have any cytoplasm. One of them has a lot of cytoplasm and a nucleus. The other one is just a nucleus, right? No cytoplasm. That means that there's nothing for that sperm to burrow into. That means that that first polar body that's produced there cannot be fertilized. So we have the secondary oocyte that can be fertilized, and we have a first polar body that cannot be fertilized. Okay, then the polar body and the secondary oocyte are going to start meiosis II. So they go through prophase II and metaphase II, and in that time, the chromatids separated. And one daughter chromosome went with the second polar body. The other one went with the other oocyte. Okay. But it's not. It's stuck. The other oocyte is still a secondary oocyte. It's stuck there in metaphase II. It has not been able to complete all of meiosis II. So it's stuck there, and that's what the sperm then can embed itself in, can burrow into, is that cytoplasm. So let's take a look at it on a diagram, on a picture. We'll draw it out here. FSH is what's going to trigger the start of the oogenesis. Okay? So let's just take a look here. And walk our way through it. All right, so let's, so here we have, this is the oogonium. The oogonium goes through mitosis to become a primary oocyte. Okay, so now we have a primary oocyte. Each one has 23 pairs of chromosomes, right? And each one is going to go through meiosis. So this is still before birth. Before birth, right? So the primary oocyte starts the process of meiosis 1. Okay, so if you remember, let's see if I can get okay. So we have 23 pairs of chromosomes here. So here's paternal, here's maternal. That's a pair because they both code for the same exact thing, right? They're both coding for the same thing. All right, so in meiosis 1, we had a couple of things happening. First of all, before meiosis 1, we get DNA replication. And we get tetrad formation. Tetrads form. Okay. So what does that mean for us? So I'm just going to do one of these and not the other one. What that means is that we have 2 of those now because we replicated. 2 of those now because we replicated. And they're linked together, right? And then those become a tetrad. So now we have and they're all linked together like one big happy family. Right? So everything's linked together. So this is prophase 1 here. We get our little crossing over effect, so we'll get some blue over here, we'll get some pink over here, and some pink over here, so we get all our crossing over, and then that's where it stays. That's boom, that's it, that's what she's born with, right? So what do we have in there? You know, it's just stuck in there, so it's a cell with a lot of chromosomes. It's 23 pairs plus their copies. I mean, there's a lot of chromosomes in there. But this is what happens before birth. This is all before birth. And it's stuck there. Okay, then she hits puberty. Right, puberty. And in puberty, meiosis 1 finishes. Meiosis 1 completes. Okay. So what happens at the end of meiosis 1? After that divides, we get 2 cells. We get the maternal cells over here. We get the paternal cells over here. They're linked together. There's 23 of those, so we have 23 chromosomes. 23 chromatids is what we really have, which means we have 23 chromosomes plus their copy. This one has 23 chromatids, this one has 23 chromatids. Okay? That's meiosis 2. meiosis, that's the end of meiosis 1. And now meiosis 2 is going to continue. So each of these are going to end up going through this same process, okay? And in meiosis 2, these things just divide. So if meiosis 2 were to go all the way through, we would end up with 23 chromosomes in each cell. Okay? Now, first of all, only one of these cells is going to have cytoplasm. Only one of these cells is going to be viable, right? The reason for that is, at the end of meiosis 1, one of the end of meiosis 1, one of these cells will have no cytoplasm, so it is called a first polar body. The first polar body goes through meiosis 2, and since you didn't start with any cytoplasm, you're going to end with no cytoplasm. And those are called second polar bodies. Also, in the process of meiosis 2, at the end of meiosis 2, or even as it splits, right when it splits, one of those is also a polar body. So we end up at the end with 1, 2, 3 polar bodies and 1 viable oocyte. Now we never really got this far because in meiosis 2 we're just stuck in metaphase. Metaphase 2.
So that's a viable secondary oocyte?
Yes.
Okay.
Yep, it's a viable secondary oocyte. And that's what gets released during ovulation. Okay. FSH has to trigger this whole thing at puberty. So the girl is going to have a significant rise in FSH. And that's what's going to cause her to complete meiosis 1 and continue with meiosis 2. Okay, now the other thing that's interesting about the female is not only do we have the development of the oocyte, we also have the development of the follicle cells surrounding the oocyte that nurture and help the oocyte go through this process. Like in the male, we have the sustentacular cells, the nurse cells. That's where the spermatogonia were going through spermatogenesis. The female is going to have follicles surrounding that developing oocyte. Okay, so she starts out with these primordial ovarian follicles, and that's surrounding the primordial ovarian follicle surrounds the primary oocyte. And then once a month, that primordial follicle, FSH, is going to stimulate that to start developing. So then the primary ovarian follicle, the primordial follicle develops into a primary ovarian follicle. From there, the primary follicle will develop into a secondary ovarian follicle. And then from there, it will develop into a tertiary ovarian follicle. Okay? The tertiary ovarian follicle is otherwise known as the Graafian follicle, and that's the one that's going to kind of bulge out and burst through that ovary, the walls of the ovary. So we can divide this phase. We're going to look at the ovarian cycle that talks about how the ovaries and the oocytes develop together. And we can divide it into a follicular phase, ovulation, and then a luteal phase. So the follicular phase, let's look at that. Here you have a bunch of primary ovarian follicles. So they're just a bunch of cells. You know, here's the primary oocyte right here, and the primordial ovarian follicles are surrounding that primary oocyte. Okay. Several of those every month get stimulated by FSH to start developing, right? So several of them start to develop. And those primordial follicles develop into a primary ovarian follicle. Primary ovarian follicle. Now the primary ovarian follicle, so this is primordial. The primary ovarian follicle still contains a primary oocyte. Okay, primary oocyte. Right? And this is a primary follicle. The primary follicle then develops into a secondary follicle. And that secondary follicle still contains the primary oocyte. Okay. The secondary follicle develops into a tertiary follicle, otherwise known as a Graafian follicle. And now we've finally completed meiosis 1, and now we have a secondary oocyte. Okay. So we went from a primary follicle, primary oocyte, primordial follicle with a primary oocyte, a primary follicle with a primary oocyte, a secondary follicle with a primary oocyte, and then to a tertiary follicle with a secondary oocyte. So meiosis 1 completed. The secondary oocyte is in the tertiary ovarian follicle, starting to go through meiosis 2, but gets stuck in metaphase 2. And then the Graafian follicle, the tertiary follicle, it's getting so big, it's pushing against the wall of the ovary, and it bursts. And that secondary oocyte is released. So what happens after that is we still have the follicle cells, right? And during this time, those follicle cells here, as the ovarian follicle was being developed, as it's developing, it's starting to produce estrogens. So we have kind of a low level of estrogens being secreted here. And the low level of estrogens is inhibiting LH from the brain from being released. Okay. It's preventing it from being released. But then we get to this point where it's a tertiary follicle, and those cells start producing a larger amount of estrogens. And the larger amount of estrogen will stimulate LH to be released. So LH gets released, which is called luteinizing hormone, and that causes the tertiary follicle to erupt or to burst, and the secondary oocyte gets released, and we call that ovulation. Then the follicles are going to do something. The follicles start to degenerate a little bit into this yellow body, which we call the corpus luteum. And the corpus luteum is going to continue to produce some estrogens, but mostly what the cells do is they produce the hormone called progesterone. And progesterone will be released and stay high for about a week. Progesterone is going to keep the uterine lining thick, so in case that secondary oocyte gets fertilized, the endometrium in the uterus is ready for it. The progesterone will keep it thick. Okay? If it doesn't get fertilized, if the secondary oocyte doesn't get fertilized, the corpus luteum then degenerates into what's called the corpus albicans, a white body. And the white body can't produce estrogens and progesterones, so estrogen and progesterone levels plummet. Okay. When they plummet, that causes the uterus to slough off, to shed. Also, when they plummet, that triggers the hypothalamus to start producing gonadotropic releasing hormone, which stimulates FSH to be released, which starts this whole process over again, because FSH caused these follicles to start developing. All right, so let's look at the uterine cycle, and then we'll kind of try to put both things together. So the uterus has its own cycle, and it coincides with the ovarian cycle. And the ovarian cycle is going to be about 28 days, and the uterine cycle, or menstrual cycle, is going to be about 28 days at the same, about the same. They're the same. Ovarian cycle controls the uterine cycle, okay? There are some terms here. Menarche, that's your very first uterine cycle that a female might have. The very first one that she has where she starts bleeding. That's going to usually start at around age 11 to 12. And then when she stops bleeding, that's called menopause, and that's where her uterine cycles have stopped. And that's usually between the ages of 45 and 55. There's 3 phases to the uterine cycle. There's the menstrual phase, the proliferative phase, and the secretory phase. So this is all about the endometrium. The endometrium is going to get thick, the endometrium is going to secrete, the endometrium is going to shed. All right, so it's 3 different things that happen. So first we have menstruation. That's where we usually start. Day 1 is day 1 of bleeding in a female. The first day that she's shedding that endometrial lining, that's day 1 of her uterine cycle. That's also day 1 of her ovarian cycle. Okay? So during the menstrual phase, she's shedding off the uterine lining, the endometrial lining, and it's beginning the ovarian follicular phase. Okay. So go back and look at this, because I don't know that I actually told you the names of it. The follicular phase, so this is ovarian development. This is the ovarian cycle. This is follicular phase because we're developing the follicle. This is ovulation because the secondary oocyte is being released. And then this is the luteal phase, when the follicles become the corpus luteum and then the corpus albicans. All right, then coinciding with that, the menstrual phase, like I said again, that is day 1 of bleeding. most of the time that lasts 1 to 7 days. Menses, the bleeding portion of it, where the endometrial lining is shed, lasts 1 to 7 days. About 35 to 50 milliliters of blood are lost. Only the functional layer is affected. So most of that functional layer sheds off, except for probably just one layer of it, a couple layers of it. The deep layer, basilar layer, is not going to shed off. Dysmenorrhea is just a term that's used for painful menstruation, and there's several causes to that. All right, so let's talk about the different phases. The first phase of the uterine cycle is called the proliferative phase. Okay, so that's where the estrogens that are produced from the follicle cells start making the endometrium proliferate. And proliferate means stem cells are dividing, going through mitosis. That lining's getting thicker and thicker. Blood vessels are forming because we have extra tissue. We need extra blood vessels growing up into that area. It completely restores the functional layer. By the time you're done with that proliferative phase, that lining is as thick as it was the previous month before it shed. Okay, it's going to, it's going along at the same rate as the follicular phase in the ovary. And so at the end of the proliferative phase, the ovary will have reached the tertiary phase, where we have the tertiary ovarian follicles. Okay. So at the end of the proliferative phase, we have the tertiary follicles with the secondary oocytes. Okay, the proliferative phase is going to last. It's sustained by the estrogens. The area is very highly vascularized, and then we end up going into the second phase. The second phase is called the secretory phase. So the secretory phase begins right at ovulation. So in the ovary, we had the follicular phase, and we had the proliferative phase going together. Now we're at ovulation in the ovary, and in the uterus, we're in the secretory phase. In the secretory phase, uterine glands start to enlarge, and more secretions are produced. Okay? As long as the corpus luteum remains intact, the secretory phase will continue on. The only way that the corpus luteum is going to stay intact long term, is if the egg gets fertilized, if the oocyte gets fertilized. Secretion peaks about 12 days after ovulation. We know from ovulation until the very next period, it's going to be about 14 days. That's where we come up with a 28-day cycle. 14 days are in the secretory or the proliferative phase, and then we have another 14 days in the secretory phase. So, okay, and this phase is going to end as the corpus luteum stops producing hormones, stops producing progesterone, as it starts to degenerate into the corpus albicans. All right, so the reproductive cycle in the female is controlled by the pituitary gland, as well as the gonads, as well as the ovaries. In the pituitary, we have FSH and LH being released. In the ovaries, we have estrogen and progesterone being released. So both of those things are going to control all of these things going on in the female reproductive system. Let's look at the gonadotropic releasing hormone. So the gonadotropic releasing hormone has kind of a pulse. It has a pulse of what hormones. The hypothalamus has this pulse of releasing the gonadotropic releasing hormone. And when it's at a lower pulse, a slower pulse, it's going to inhibit hormones. When it's at a higher pulse, it's going to stimulate it. So it's affected by both estrogens and progesterones. So estrogens will increase the frequency of gonadotropic releasing hormone being released, whereas progesterone will decrease the pulse frequency. Okay? So estrogens increase the pulse frequency and progesterone decreases the pulse frequency. So let's look at this. Early in the follicular phase of the ovarian cycle, so the ovaries, in the ovaries, the follicles are changing from primordial to primary to secondary to tertiary follicles. Early in that, from primordial to primary to secondary, the estrogen levels are low. They're not going to get high until that tertiary follicle is produced. So when it's low, that means the gonadotropic releasing hormone frequency is going to be low, 16 to 24 per day. Estrogens are low, gonadotropic releasing hormone is low. Okay? As the tertiary follicles form, the estrogens rise really steeply because all those follicle cells are releasing estrogen. The estrogen inhibits luteinizing hormone. We don't want luteinizing hormone released too suddenly because luteinizing hormone is going to cause ovulation. We want that follicle to completely develop. We want that oocyte to completely develop into the secondary oocyte and halt in metaphase 2. Then the pulse frequency of the gonadotropic releasing hormone is going to increase to 36 per day. Okay, so it's going to rise. As estrogen levels rise, gonadotropic releasing hormone is going to rise. The increased pulse frequency of gonadotropic releasing hormone is going to stimulate LH secretion. LH secretion then is going to cause ovulation. We're going to take a break here real soon. I'm going to get you a worksheet to work on. Okay, so usually,
Which hormone stimulates the secretion of LH?
it's actually just the frequency of the gonadotropic releasing
Okay.
hormone that does that. So estrogens, as soon as they rise really frequently, then gonadotropic releasing hormone rises
Okay.
it starts pulsing more frequently, and that stimulates luteinizing hormone to be released. That's going to happen around day 10. Okay, so gonadotropic releasing hormone starts increasing its pulse frequency at day 10. 4 days later, ovulation occurs. Luteinizing hormones released in high enough numbers that ovulation can occur. So remember, estrogen, at first it was at a low level, low pulsing, low level of estrogen. Then we got to high pulsing, high level of estrogen. So estrogens flipped. At low levels, they inhibited luteinizing hormone. They steeply climbed, and at higher levels, they increased luteinizing hormone, right? So it's just a matter of the low levels inhibit it, high levels increase it. And then you get the massive ovulation. All right, so let's take a quick break. I'm going to go print out something